Hello guys, I am Dhruv Shah from BTech third year. This video is prepared in the partial fulfillment of the requirement towards the project of the course SC462 Elements of Synthetic Biology. The name of our course instructor is Professor Manish K. Gupta. In this video, I will be talking about the powerful genome editing tool CRISPR. I will be explaining how CRISPR is creating revolution today by taking Retinitis pigmentosa as an application of CRISPR. CRISPR today has emerged as a very powerful genome editing technique. So, first of all, I would like to give a brief introduction of CRISPR. The power of CRISPR can be very well illustrated by giving example from our day-to-day lives. Talking about the field of medicine, in particular, CRISPR has contributed in many ways. CRISPR has helped in finding a solution for stopping cancer cells from multiplying. Scientists have also got success in reversing mutations that were the cause for blindness. CRISPR has enabled scientists in making cells impervious to the virus that caused AIDS. Well, these were the success stories in the field of medicine. CRISPR has also helped agronomists in finding solution to killer fungi which destroyed the wheat. CRISPR has been successful in altering the DNA of yeast, thus producing ethanol. This will bring an end to a dependence on petrochemicals. This was all about CRISPR. But what actually is CRISPR? When viruses infect a cell, they inject their DNA. In bacterium, the CRISPR system allows the DNA to be plucked out of the virus and inserted in little bits into the chromosome, which is the DNA of the bacterium. These integrated bits of viral DNA get inserted at a site called CRISPR. CRISPR is an acronym which stands for Clustered Regularly Interspaced Short Palindromic Repeats. It is a mechanism that allows cells to record over time the viruses they have been exposed to. Once those bits of DNA have been inserted into the bacterial chromosome, the cell then makes a little copy of the RNA. These little bits of RNA from the CRISPR locus associate, then they bind to a protein called Cas9 and forms a complex that functions like a sentinel in the cell. It searches through all of the DNA in the cell to find sites that match the sequences in the bound RNAs. When those sites are found, the complex associates with that DNA and allows the Cas9 cleaver to cut up the viral DNA. It makes a very precise break. The Cas9 RNA sentinel complex can be thought of as a pair of scissors that can cut DNA. It makes a double-stranded break in the DNA helix. The CRISPR-Cas9 system is an RNA-guided nuclease system for the targeted introduction of double-stranded DNA cleavage. Targeted genomic cleavage requires two key elements. First, a homing device, and second, an endonuclease. In the CRISPR-Cas9 platform, the homing device is guided RNA, gRNA, and a Cas nuclease. This diagram depicts how CRISPR mechanism works, which I have explained before. The scissor plates shown here represent the Cas9 enzyme. Something more about CRISPR. The gRNA contains a 20 nucleotide target sequence immediately upstream of a PAM, protospacer adjacent motif. This is sufficient to direct the Cas9 nucleus to the complementary site in the genome and create a double-stranded break. This is analogous to the way we use a word processing program to fix a typo in a document. Cells have the ability to detect broken DNA and repair it. So a cell can fix the break either by pasting together the ends of the broken DNA with a little change in the sequence of the position, and second, by repairing the break by integrating a new piece of DNA at the site of the cut. This diagram represents CRISPR and its functioning. Genomic DNA, how the CRISPR matches the genomic sequence, the PAM sequence, the guide RNA, and the Cas9 protein. In the beginning of the video, I have discussed the application of CRISPR. Some of them, some other applications of CRISPR are today many startups devoted especially to CRISPR have been launched. CRISPR research and development has been spun up by many international pharmaceutical and agricultural companies. CRISPR and the disease retinitis pigmentosa. Just as other applications of CRISPR, treatment of retinitis pigmentosa is a very recent breakthrough in the field of medicine. The motive of the entire process is to appreciate the use of CRISPR as a powerful tool for genome editing. Genome editing in induced pluripotent cells has been successfully demonstrated to be very effective for generating disease models for genetic disorders. Something about stem cells. Okay. Stem cells are special types of cells with remarkable potential to develop into many different types of cells during early stages of growth and life. These stem cells serve as the internal repair system for many tissues of the body. These stem cells help to replenish other cells by dividing essentially through a process called mitosis. The new cell thus formed has the potential to remain a stem cell, or it can also change to another cell with more specialized functions. This diagram depicts the various locations of somatic stem cells in the body. They are found in the brain, skeletal muscle, peripheral blood, heart, skin, etc. Stem cells. Stem cells basically possess two distinct characteristics which help them to differentiate from other types of cells in the body. First one is self-renewal. Their ability to renew themselves by going through several cycles of cell division and still preserving their undifferentiated state. The second property is called potency. Their capacity to be induced and hence be differentiated to become organ or tissue specific cells with special functions. So the two distinct properties of stem cells are described here. A single cell that can replicate itself, and at the same time it has the ability to differentiate into many cell types. Now I will be explaining about induced pluripotent stem cells. Scientists have been mainly working on two categories of stem cells, namely embryonic stem cells and non-embryonic somatic or adult stem cells. In the year 2006, scientists got a major breakthrough by establishing the conditions that would allow genetic reproduction of some specialized adult cells so that they could assume a state like that of stem cells. They are called induced pluripotent cells, and it is our topic of concern. Formation of induced pluripotent cells. The adult fibroblast cell is reprogrammed and thus results in the formation of induced pluripotent cells. These are the various iPS cells: cardiomyocytes, adipocytes, neural cells, pancreatic beta cells. Okay. Induced pluripotent stem cells are a category of stem cells derived from adult cells that have gone through genetic reprogramming to achieve a state like that of an embryonic stem cell. It was the iPSCs of mouse that were first reported in the year 2006. Then later in 2007, human iPSCs were reported. Human induced pluripotent stem cells can be generated from two sources, namely human fibroblasts and human renal epithelial cells in urine. So the mouse iPS cells were reported in the year 2006, and later in 2007 human iPS cells were also reported. Now I will be describing about the disease retinitis pigmentosa. Retinitis pigmentosa. It is a rarely occurring genetic disorder involving breakdown and loss of cells in the retina. With the disease retinitis pigmentosa, photoreceptor cells in the retina called rods and cones die. The affected people start losing their visual field. Such people face difficulty in performing their day-to-day activities like writing, reading, and walking without assistance or identifying objects and faces. So this diagram represents the comparison of normal eye and eye with the affected with retinitis pigmentosa. Okay, so the eye with retinitis pigmentosa has pigment deposits known as bone spicules, and this represent the pigment clumps. Okay. Effect of retinitis pigmentosa on vision. The diagram on the left shows the normal vision, and that on the right shows the vision with retinitis pigmentosa. You can clearly see that the disease has affected the vision to a much larger extent. Effect of retinitis pigmentosa on the retina. The left shows the normal retina and that on the right shows the retina affected by retinitis pigmentosa. You can clearly see that the rods and cones of the retina are affected by the disease. There are in total 3 ways in which the disease can be inherited. They are autosomal recessive inheritance, autosomal dominant inheritance, and the third one is X-linked inheritance. Out of these 3 ways, the third way, that is the X-linked inheritance, is a topic of concern. Fixing of the disease by CRISPR. Induced pluripotent cells generated from patients' fibroblasts can be used as a potential source of autologous cells for transplantation in the disease. But the problem here is that iPSCs derived from patients would still continue to have the mutations which were responsible for the disease. This is where the genome editing tool CRISPR comes into play. The motive of the scientists was to repair these mutations with CRISPR and thus generate patient-derived cells which were healthy. The scientists thus checked if CRISPR could be used in patient-specific iPSCs to fix X-linked retinitis pigmentosa by repairing the RPGR point mutation. Fibroblasts were cultured from a skin punch biopsy of a patient suffering from the disease. Next, they were transfused to generate iPSCs carrying the mutation of the patient. For this purpose, CRISPR guide RNA and Cas9 endonuclease were used. 13% of retinitis pigmentosa GTPase regulator gene copies showed conversion to the wild type allele and mutation correction. The final results. This diagram shows the clinical findings and iPSCs from an adult male XLRP patient. In this diagram, the procedure for validating the guide RNAs for gene targeting is shown. The last diagram represents the correction for the RPGR mutation in patient iPSC line. Two major results of the study performed by scientists are as follows. The first one, deep sequencing of transfected cells showed that 13% of reads contained precise correction of the mutation with replacement of the premature stop codon tag by the wild type GAG codon. Correction of mutation was not seen in any untransfected control iPSCs. So these are the two major results which the scientists got from the study. I hope you enjoyed watching the video. I hope the video has helped in arousing curiosity about CRISPR and understanding about its immense power. The link to the presentation used for this video is present in the description of the video. Thank you.